Welcome to the Chapter 18 review. So Chapter 18, remember, that one covered the diagnosis of infections. So there were a whole bunch of tests that we went through, basically. We have the phenotypic, immunologic, and genetic tests. We went through a few little random ones as well. Steps involved in testing your patient. Well, first you want to get a specimen, an appropriate specimen from the appropriate location, using appropriate media and handling it appropriately, of course. So there's all of that that you need to keep in mind when collecting a patient as part of, a healthcare team. All right, so you've got your specimen collected from your patient. You want to cultivate it now. If you do need to cultivate your specimen and grow up, and you don't know what pathogen you're dealing with, you might want to consider what kind of media you grow it on. This could include certain isolation media, which could include selective media that would it would grow some organisms, but it would inhibit the growth of other organisms. Like when we talk about McConkey agar has bile salts in it. Bile salts are going to be fine for enteric bacteria that grow in the gut, but not good at all for a gram-positive bacteria. So if you suspect it's an enteric bacteria that's causing the disease, you might put it on McConkey to keep the other stuff from contaminating. You don't want to have to isolate through all of that. You might as well just, you suspect it's this, it's more likely that it is. Let's just cut to the chase. So that's the idea with that. We also have differential media that we can use, often associated with our biochemical tests. And that differential media will have color changes based on the ability of your organism to metabolize certain things. And if it can metabolize certain things, then it will create certain products that can often cause a color change in the media that you're using. We can also utilize things like blood cultures. If you think that your patient is septicemic, you often will be drawing blood cultures if you are suspecting that. And usually if you're unsure what the pathogen is, you'll want to draw an aerobic and an anaerobic blood culture, typically, unless there's some reason to think that your patient has one or the other. Okay, so, and of course you want to be careful not to draw any air into your anaerobic, because your anaerobic organisms would not be able to survive the presence of oxygen if they are unable to deal with those toxic oxygen molecules, right? Because they're not trained, they're not prepared for that. Okay, biochemical tests. Again, your organism will be able to metabolize certain molecules. That's probably going to be different from other organisms and what they can metabolize. And then based on your results from certain biochemical tests, you can compare those to a dichotomous key or something similar to a dichotomous key, like a computer program that is made to analyze a large array of all these biochemical tests that you can do all at once, that you put into the little bubble system, and it changes a color or not. And based on whether it's positive or negative for each test, you have a number that it will tell you how to interpret. And you plug it in and you get that your answer is E. coli. So that is how the biochemical tests often work today. But yes, it's the same thing as a dichotomous key. It's just that we have a computer doing that for you. A dichotomous key, basically, like you would do a test, and let's say it's gram-positive or gram-negative. And if it is gram-positive, then let's see if I have one over here really quick. Yes, I do. You are lucky. Okay, so let me show you a dichotomous key just so we can run through it really quick, because I don't want this to be confusing for sure, because you have to do this for your unknown stuff. So here is an example of a dichotomous key. You don't have to worry about too much about how the breakdown is on this, but let's say that you had a gram-negative organism. This says gram-negative. Okay, and let's say you're gram-negative. When you grew it in your fermentation media that had your different kinds of sugars in it, you did not produce gas in your glucose. So did you produce acid in your sucrose now? Let's say you did. You did produce acid, this one here. All right, next, did you produce indole? Well, you did your SIM media, right? And you added your Kovacs reagent on top and it turned red. So it is indole positive, which is there. And then that only leads to one thing, and that is Proteus vulgaris. So that is the organism that your patient is sick with, turns out. Okay, so that's how that works. So it's actually pretty nifty, if you ask me. And it is somewhat useful for situations where you don't have the information or the means to do other testing like PCR or something. So biochemical tests are actually useful. Next, we have antimicrobial sensitivity. That's like Kirby-Bauer. Doing the Kirby-Bauer can tell you not only what antibiotic you should be using to treat your patient who has the bacterial infection, but it will also tell you potentially, depending on the organism, it could potentially tell you what genus of organism you have based on the profile of the results that you get from the Kirby-Bauer assay. We also have phage typing. Phage typing is using the concept that phages, bacteriophages, which are viruses that infect bacteria, that these guys only infect certain species of bacteria or even strains of bacteria, depending, but it's always very specific. So we'll take a phage that we know is associated with salmonella. If we think the patient has salmonella, we'll take their sample that we collected and isolated from their stool sample and have spread it across this whole Petri dish and dot it with certain phages. And we expect that in the little area where we dotted a sample of bacteriophage that infects only salmonella, maybe salmonella typhi, which causes typhoid fever. Let's say that there's a clearing there. Well, those are called plaques, and that's because the phage killed all the bacteria in that little area, because the phage was able to infect the bacteria in that area. And it only infects certain bacteria. So that means that you have salmonella typhi as your cause of your patient's illness. So that's how you would use phage typing. For certain immunological tests that we looked at, we looked at all sorts of serological tests that involve a patient serum, which nowadays it doesn't only deal with patient serum. That's just a similar concept. The original concept of where the antibody would come from, but we can now use other bodily fluids. So that's serology, getting antibody or antigen from a patient sample and using an antibody antigen specificity reaction in order to determine the identity of things. We looked at certain different kinds of in vitro testing, including the Western blot, where we run it on a gel, move it to a membrane, and then, put the patient serum mixed on there. It gives us where we can see the band. That means the patient had the antibody, and that means they're positive for what the band is, HIV, okay? Then we have immunofluorescence, where we have antibodies that are linked to some sort of fluorescent molecule that glows. And if that antibody can associate with the thing, then we would associate that with the presence of that thing. So that could be looking for the antigen in the patient, in which case that is the direct version of immunofluorescence. However, indirect version of immunofluorescence, what we would have would be a known antigen put on a substrate. Then we would add the patient serum, which we think might have the antibody. And if it does, we'll add another secondary antibody that binds to antibodies. And it has the fluorescence on it and will glow because of that. So that's how you do direct versus indirect, respectively, for the immunofluorescence. With ELISA, which is enzyme-linked immunosorbent assay, we have stuff sticking in the bottom of a plastic well. And essentially, it's going to be either antibody that's stuck down there or antigen, one or the other. So what we'll do is we will put a known antibody down there, put the patient sera in there to see if the antigen binds to it. And if it does, we'll put another antibody in there. It has an enzyme on there. It reacts with a substrate that causes a color change. If the enzyme is there to interact with the substrate, that means it's positive. That's the direct method, the sandwich method. Then we have the indirect method of ELISA, where we want to know we want to know if that patient has the antibody, and it works very similar to what I just explained to you with the immunofluorescence. In this case, we have antigen in the well, and put the patient serum. So it comes in, it binds to the antigen that we know, our unknown patient serum binds onto it. And then we'll add a secondary antibody and it'll bind to that other antibody. And it has an enzyme and it's going to cleave the substrate and it's going to cause a color change. And that's positive. That's the indirect ELISA. That's what we did in the lab, in our lab exercise. Okay. So a lot of these tests, you might want a secondary test in order to confirm your results because they might not be super specific. Right. So anyways, a lot of times with ELISA, if you have a positive on the ELISA, we will run a Western blot to confirm. And that's important because we do actually do that in real life. Whenever we test for HIV, we will run the Western blot if the ELISA suggests it's positive. So we can confirm it that way. All of these, all right, let's go complement. Complement, essentially we can use that concept of how complement works to see if a patient has an antibody to something. I am not going to sit here and explain it out again because I just feel like it's a lot of steps. But basically what it works like is if you have that antibody and I mix antigen in there and they bind together and then we introduce complement, it gets stuck to that. So now when we add red blood cells, the complement here, that guy that would normally lyse and break open the red blood cells, that guy's it's stuck with this. It can't do it. So the red blood cells stay intact if you are positive. Otherwise, a negative test, you would have bursting open of it because there's no antibody to conglomerate everything up and bind everything up. So that's the idea of how that works. It depends on the function of the membrane attack complex done by complement, right? Complement cascade. So those all depend on specificity versus sensitivity. Specificity, you're less likely to have false positives. Sensitivity will detect every little positive. So specificity, you're not going to have any false positives. You're only going to bind to the thing that you're interested in, and only the thing that you're interested in. But you might miss a few. So that would give you a problem with false negatives, but you wouldn't have any false positives. Sensitivity, if you're super, super, super sensitive, you might be overreactive too. So you could get false positives. So you detect every positive, but some of them might be fake. So there's an issue there that isn't always the case with all of these, right? But those are risks that you take in certain circumstances when we're considering specificity versus sensitivity of these tests. Now, moving on to the genetic testing, we talked about PCR, which is polymerase chain reaction. That is DNA amplification. There's a lot of different versions of it available now. RT-PCR, real-time PCR is the most common. We have hybridization assays where we're going to have probes of known DNA sequences that we can match up to a patient sample. And if it does bind and have fluorescence again, that would allow us to determine that, oh, you do have this sequence present in your serum. That means that you also, hey, by the way, hate to break it to you, have, I don't know, salmonella in your serum. I hope you won't have that in your serum, but you know what I mean. Then we have a microarray, which is a similar concept to that, but we have a whole bunch of different stuff dotted all over this different plate. And then we treat it with the patient serum. And that if we have a DNA hybridization going on there after that, means it'll bind at specific points all over the plate or just in one point on the plate, depending on what kind of plate you're using. And that would allow us to determine if it is likely that you have this disease versus this disease, but it's a pretty quick assay. And yeah, it's useful that way if you have known probes available for these tests. And then we have mass spec, which it's like a MALDI-TOF machine. Basically what it does is shoot lasers at your sample and ionize it. And then their sensors pick up those ions that you make from doing that. And based on the pattern that it, order that it picks up or how it behaves, then that's how it determines what your sample is made of, literally down to the atom. And that can, everything has its own little profile. So it can diagnose things that way. And it can also determine, antibiotic resistance that way. And then imaging, of course, to look at certain areas of the body, if you have localized infections or deep tissue infections, something like that, where we can assess our, approach without actually having to cut people open. So it's less invasive and safer for the patient. So that is it for chapter 18. I hope that was fun for you because it was for me, and I will see you guys for the next one. Bye-bye.